Hi everyone. The Riodinidae family contains about 1,500 species, which are included in nearly 150 genera. These butterflies, popularly known as metalmarks, are found worldwide. 90% of their species are restricted to the neotropical realm. This contrasts with its poor representation on the Caribbean islands, where only one extant genus is known. The genus Dianesia is monotypic and is composed by the nominate subspecies Dianesia carteri from Bahamas and subspecies rhamdeni, which is found in Cuba and was described from Baracoa on the east. These remain one of Cuba's least known butterflies, about very which little information is available. Several scattered populations are found across the island, and it is said to have crepuscular habits and to perch on the underside of leaves, which is a common trait of the family. Nunez and Barrow 2016 classified it as vulnerable to extinction according to UCN criteria because of its fragmented distribution and its vulnerability to anthropogenic threats. Very recently, Esnar and collaborators 2023 described part of the life cycle of this butterfly and recorded 4 species of the genus Buxus as hosts of their larvae. The genus Buxus has the biggest world radiation center in Cuba, with 37 species, of which 35 are endemic, and most of them are exclusive to the mountains of northeastern Cuba. These plants are known for their ability to sequester heavy metals from the serpentinic soils that most species grow at, and because there are several Cuban species threatened of extinction because of habitat loss. Several authors have pointed out the existence of polar morphs in the Cuban populations of Dionysia. For example, Alayo Hernandez 1987 said that the population from the southern coast of Santiago de Cuba was a light morph typical of dry habitats when compared to the darker mountain specimens of Baracoa. On the other hand, Hernandez and collaborators in 1988 say that the specimens from the northern keys belong to the same population of Bahamas, the dominant subspecies. This would imply that both of them live in Cuba. Finally, these authors collected the aberrant specimens in 1976 on the outskirts of Baracoa City, which was different from the specimens of typical Dionysia carteri flying there. Whether this variability is the result of phenotypic plasticity or a cryptic diversity has remained as an open question ever since. The possibility that this genus contains several cryptic species will have deep implications for conservation, since most of those species could be represented by isolated small populations which will be very vulnerable. As a first approach to address this research has the objective of reconstructing the phylogenetic relationships of Dionysia, employing evidence of molecular data, morphology, and natural history. We conducted fieldwork on all the known populations of Dionysia in Cuba, Lomas de Galindo in the west, the northern key center of the island, and the Nipisagua-Baracoa Mountains on the east, where the butterfly was described before. For the first time in 70 years, we rediscovered the populations of the Cienfuegos Bay and the southern coast of Santiago de Cuba. We also documented more specimens of the putative aberrant morph of Baracoa. Using the distribution of the host plant, we located 2 new populations, one of them in the Serpentine Hills north of Forkin City on the east, and in Santa Clara in the center. Given that the butterfly was poorly represented in public collections, we collected specimens to perform both morphological and molecular analysis and also documented the larvae and host plants of each population. DNA extraction, sequence amplification, and sequencing follow standardized protocols for butterflies. We've sequenced both the barcodes, a fragment of the COI gene, and the whole genome of the butterflies employing the low coverage technique. We calculated pairwise distances employing the software Mega11 and reconstructed the phylogenetic relationships employing the maximum likelihood and Bayesian inference analysis on IQ3 and Mr. Bayes, respectively, in the Cypress platform. Thanks to staff of the McGuire Center for Lepidoptera Research, we were able to obtain sequences of the Bahamian population, and we also retrieved additional sequences from other Riodinidae genera from GenBank to conduct a rooting of our trees. Our trees were visualized employing the software Fig3, and we also employed the software Assemble Species by allopatric partitioning to conduct species delimitation analysis. Both the maximum likelihood and Bayesian trees obtained similar topology, so we only showed the Bayesian trees which had a little bit of better support. In all scenarios, all these 9 population systems of Dionysia were recovered as reciprocally monophyletic clades except for 2 of them. The aberrant population is the first one to diverge, and from the outside to the inside, we have clades composed by the Bahamian population, the Cienfuegos Bay 1, the Nipisagua-Baracoa 1, the Holguín 1, and a polytomy, which is composed of the Santa Clara population, the Lomas de Galindo population, and a single clade that contains all specimens of the Northern Keys and the Santiago de Cuba population nested as a single branch inside of it. The whole genome sequencing tree, for which we have the preliminary results, since we don't still have genomes of the Cienfuegos and Santa Clara population, shows a very similar tree topology with two major changes. First, the Holguin serpentines population is diverging first than the one from the Nipesawa Baracoa, and the Santiago de Cuba population, which we still have represented with only one genome, is sister to the population containing all specimens of the Northern Keys population instead of nested inside. Peru's instances changed from 1.59 from the Northern Keys and Santiago de Cuba population to 7.08 between the aberration and the Cienfuegos Bay population. Except for the Northern Keys and Santiago population, all other species and populations show genetic distances well above the 2% that is usually employed as a threshold value for barcode gaps in Lepidoptera, indicating that these could be considered different species. The species delimitation analysis recovered 10 species hypotheses ranging from 7 species to 21. The best score hypothesis recovered 12 species, which are the 9 population systems and clades that we talk about on the first three, including the Santiago de Cuba and Northern Keys population as independent clades, and 3 additional species. These species, in the case of 2 of them, were composed by specimens collected in the Nipesawa Baracoa Mountains, where older specimens population were found, and the third species was composed by a single specimen which was collected on the outskirts of the distribution of the Northern Keys population and was sister to it on all the clades. These specimens belong to species that are more widely geographically distributed and seem to have a bigger genetic variability. So they should be considered as specimens with slightly different barcodes that were covered as different from the analysis, and we stick to the 9 species that we mentioned before. As we previously said, the barcodes recover the Santiago de Cuba and Northern Key population as a single clade. However, these populations show very important morphological differences. For example, in the case of the Santiago de Cuba, we can see a darker background color, a greater extension of the posiscal white band, and in the case of the larvae, broader black lateral spots and more marked white dorsal lines. Despite barcodes being used as a popular tool for species delimitation, sometimes they fail to recognize species of recent origin, and they cannot be used to construct robust phylogenies. However, the differences that we saw on these morphological traits, the results of the species delimitation analysis, and the preliminary results of our whole genome sequencing indicates that these could be considered different species as well. All of the populations of Dianesia exhibit differences in their morphology regarding the background color, the existence, shape, and distribution of the white spot on the postdiscal band, and other minor details. The larvae also differ on the background color, the extension and position of the black lateral spots and white dorsal lines. These butterflies, these differences have been tested employing both color pattern analysis and morphometric analysis, which we do not discuss because of the limit, but also help to distinguish them. Host plants that populations use also differ. Buxus gonoclata is used in Galindo, Santa Clara, and Holguin in serpentine soil. Buxus glomerata is used in Cienfuegos and Santiago in karstic soil. Buxus bahamensis is employed on the Northern Keys and likely on the Bahamas, also on karstic soil. And Buxus marginalis and Buxus schaefferi are employed by the aberrant and Nipesawa baracoa species. The last one is also polyphagous and is known to employ at least 10 species of Buxus as hosts. Habitats in which these butterflies inhabit are very different as well. They range from lowland or mountain, very humid rainforest to lower serpentine or coastal dry scrub woodlands. These habitats seem to be a perfect scenario for the diversification of these butterflies, which have a poor dispersal power since they are poor flyers, and this may have helped them to radiate on the island. So we conclude that the genus is composed at least of 9 species: Dianesia carteri from Bahamas, Dianesia transdini from northeastern Cuba, and 7 new species which were recently described on a paper that we published on SOTAC. The future goals of this research are to evaluate new populations that we are discovering. They might represent additional populations of these identified species or new species which may await description. We will conduct population genetics to the species with a broader range to understand how the genes change across the landscape. We will try to understand why these butterflies may prefer a particular host plant or not. We will study the biography, how they origin and evolve, and how to use distribution modeling to identify potential areas for the distribution. We will conduct a study on the heavy metals that they may uptake from the leaves, and we will work on a UECN assessment for these species. 6 of them are currently being evaluated, and they all seem to be at some sort of endangered category. I would like to thank my work team, our team Dianesia, who has been working very actively with me on the field to collect data, to process this data, and to assess all of the steps of this research, which has been a tremendous effort for my team. Thank you very much.